Thank you.
Break. Do you want to pull up your talk, since I'm a failure at this?
I'll give it a shot.
Oh, no, this is, here you are.
It should be.
It's just a matter of getting it, there,
Okay.
go for it, and then you need to be full screen.
No. Okay, so
So this is a proceedings talk, and we're now segueing into a related issue, which is the question of looking at chromosomal rearrangements. Krister Swenson.
yeah, and in order to shift gears, and I realized that I'm one of the only talks talking about rearrangements here. I'm going to take some time, 5 minutes or so, to speak about rearrangements first. And then I'll talk about the link between rearrangements and 3D conformation of the chromosome in the nucleus. This is co-work with Mathieu Blanchette, who's also in the room. So rearrangements. Here's an inversion. We have an order of genes along a chromosome, and we have 2 breakpoints. And these 2 breakpoints may get confused, and what we end up with is an inversion of the order of these genes on the chromosome. This is an inversion. These are important for many reasons. One reason is population variability. So once one of these events has happened, this will suppress recombination, for example. And what we'll end up with is many members of the population with very similar spans of chromosome. In the extreme, this can lead to speciation. So we have, it's called reproductive isolation. So we have a bunch of members of the population who can only reproduce with each other. Gene regulation problems can happen. Let's say a breakpoint occurs within a gene. This can cause a lot of problems, and serious disease can be the result. So on an evolutionary scale, people have looked at scenarios of rearrangements to transform one species into the other. This is an example on Drosophila. Things are much more complicated. In our case, we're looking at human mouse. Some of the main questions come about from. Oh yeah, sorry. Some of the main questions have come about 30 years ago, introduced by some of the people in this room, who I won't name names, but one of them is giving a talk after me. So for a while, around the turn of the century, every time there was a genome sequenced, there was analysis of the gene order evolution. The 1,000 Genomes Project, there's now an analysis within species. People call these structural variations or rearrangements. Things can be said about breakpoint reuse, the speed of chromosome evolution on parts of the tree. The basic problem is pairwise genome comparison. With this, you can rebuild species trees. You can use this information. It's a powerful information for finding orthology, parallelology relationships. And we can try to reconstruct ancestral species, or ancestral gene orders, excuse me. So this has been done many times, and it's continuing research today. But the thing is, there's there's problems with this. These are all mainly based on parsimony assumptions. And really what is happening is we have under-constrained problems here. So if we're only considering parsimony, there are many possible scenarios to transform one genome into the other. There are many possible arrangements of breakpoints. And in fact, there are exponentially number of parsimonious scenarios to transform one genome into the other. And what we need is biological constraints in order to move this along. So what's one potential biological constraint? That is the chromatin organization. The DNA is packed into the nucleus in a certain way, okay? And what we're interested in is what the relationship between this, how this is packed into the nucleus and the rearrangements that may have happened. So over the last 10 years, we've had access to high-C data. So roughly speaking, you can consider high-C data as a matrix, and an entry ij in the matrix is proportional to the proximity in 3D space that position I is with position J. So this dark spot here says that this part of chromosome is close to that part of the chromosome, closer than expected. We have information in between chromosomes too. And so we want to study that main question: is this 3D conformation related to the breakpoint regions? Are they local in 3D space? So what do I mean by local? So here are two chromosomes, a yellow chromosome, a red chromosome. It's a 2D version of that 3D picture I was showing you, and focus here in the middle. Imagine these two ends are confused. So there was some process that happened. There are many possible ways that this could have happened. That these two things were confused. This is a translocation. And this is local because they happened very close in 3D space. This has been studied in limited ways. In cancer, there are studies on translocations. They're all limited to somatic translocations. On an evolutionary level, there have been limited studies on human and mouse, human gibbon. In these two cases, breakpoints were just considered on their own in isolation. And I'll show you why that's important in a second. So far, the most, I guess, sophisticated study that has been done, it was from 2011, where they study exactly this question. They come to conclusions that, in general, indeed, the breakpoints are closer in 3D than expected by chance, and it correlates with gene-rich open chromatin regions. They only used data from human, and given that they saw this pattern using only data from human, not mouse, they concluded that there must be some sort of conservation between the two species as well. So the problem that we're trying to address in this study is that, okay, when there's no breakpoint reuse, pairs of breakpoints are easy to match up. When there's some breakpoint reuse, it gets complicated, but there are some cases you can look at. Most of the cases where there's breakpoint reuse, you lose all the information when you're approaching the problem this way. So they were able to look at less than 300 breakpoint pairs, and they didn't have enough power to look at interchromosomal pairs, so their results were all about intrachromosomal pairs. Our paper scales up this study using new methodology, and I'll show you in a second how that is, but it has to do with sampling scenarios. By sampling these scenarios, we have about 3 million pairs that we can look at. We have results for inter and intrachromosomal pairs. We have many cell lines that we are able to compare. And just to summarize the differences, so there are studies that look at a single breakpoint at a time and compare them to things like TADs and TAD boundaries and things like that. There are those that look at pairwise, and we're using new methodology that allows us to scale this up and look at interchromosomal pairs as well. So this is human and mouse. This is a cartoon of their genome. Each one of these is a genome, and each edge between two vertices is a rearrangement. So a path through this graph is a scenario. This is a parsimonious scenario. There are non-parsimonious scenarios, and our question is, perhaps there are only those scenarios that are of interest, which are those that have their breakpoints close in 3D space. So how do we look at this question? Let's take a particular scenario. Each move has 2 breakpoints. So we have a location in the heat map corresponding to each of these moves. Some are in other chromosomes, some are between chromosomes. And, well, if you draw the heat map and you put a blue dot for each of those rearrangements that we sampled, the lighter blue, the higher the frequency of that rearrangement in our set of sample scenarios. You see that the dark spots in the chromas in the heat maps roughly correlate with the blue points. And we see that also to some extent in the interchromosomal maps. But how do we make this more formal? Okay, so for each scenario now, we take the average proximity over all the moves in that scenario. We do 10,000, 100,000, as many as we want, sample these scenarios. And for each one, we bin its proximity, and it gives us this distribution. So the question is, how interesting is this distribution? Well, let's maybe assume that the breakpoint locations are not important for a second in terms of 3D space, okay? So let's randomize these scenarios in some way. So how do we randomize this scenario? Well, we take, for each move, let's say this red move, we move it to new coordinates in the genome, okay? So we have the same size breakpoint regions, we have the same distance between the breakpoints, and we do that for each one of the moves in the scenario, and we have a randomized scenario. And that, what ends up happening for the intrachromosomal maps is we end up sampling these diagonals, basically. And for the interchromosomal maps, we're kind of just randomly sampling locations. And what we see is some version of this for each cell line. So this is for interchromosomal and intrachromosomal pairs. We also permuted the data to make sure that this was that these results were significant, and we always see very significant differences between the randomized scenarios and the non-randomized scenarios. We see this for data from 3 different labs, and for data in different for cells that are in different states of their cell cycle, for cells that have differentiated in different ways. This is not a pattern that is due to just particular rearrangements. So if we look at a If we look at rearrangement length, the distance between the breakpoints in 1D, and we look at the dashed line or the actual rearrangements, solid line random rearrangements, the actual rearrangements are always closer. I don't have time to talk about the similarities between the different cell lines, but one of the interesting things about our result is that looking at the different types of cell lines and comparing the similarities between them, we actually find that the similarities are important. So this kind of leads to the question of whether selection might be occurring against breakpoints in all the differentiated cell lines and not just the zygote, which is the structure that's being passed. So to summarize, we have the rearrangement mechanisms that's probably based on being spatially close. There's possibly what we're seeing is a negative selection against rearrangements that are distanced in 3D space in each of the different differentiated cell lines. And again, we're still only using Hi-C data for humans. We have Hi-C data for other species and for two sides. There are complications with using the data for these, but that's future work. We have other results about formaldehyde concentrations in the Hi-C experiment that I don't have time to talk about. We have all the results reproducible with, you go on the website and you download the data and you can run with SnakeMake, everything just automatically is reproduced. You can substitute your own matrices. And since then, we've been working on models and algorithms to actually use this as a constraint to constrain and find more likely scenarios. And I'd like to thank Mathieu Blanchette and Anne, Fabio, Laurent for the conversations on this subject and our funding sources.
Thank you, Krister. Do we have questions about rearrangements?
Yes, excellent talk. There was a paper that shows that CTCF regions are conserved between organisms. Did you see any overlap? Where are the TADs? Where are the TADs of formation? There was a physical operation. How this interaction go about?
Yeah, there are independent results that correlate specific breakpoints with CTCF binding sites.
Yes.
So yes, there is a correlation there.
Okay.
And those would be spots that tend to be very close in 3D space. Yeah.
Yes, that can be another constraint on your model.
Absolutely. Yep. Absolutely.
Thank you.
Good point.
Do we have additional questions? That was a very nice background on the whole field, and I have a comment, which is another place where rearrangements are very important, or inversions are very important, is the formation of sex chromosomes and segregation distorter systems where one chromosome tries to take over against its sister chromosome, and the fact that there is recombination suppression allows a group of genes to act in cahoots.
Yes.
Let's thank the speaker again.